you very much. As I said, my name is Valerii Plotnikov, and I am a scientist. And I want to tell to you about the cloning of extinct animals. Sorry, my English, it's my not own language, and I will read from my preparing text. My specialization is not very closely related to the problem of modern society. But one part, even if not with my direct participation, still concern our society and the world as a whole. I am talking about cloning. I am not an expert in this field, but I work in an area that provides material for such research. I am a leading researcher of Academy of Science of Sakha Republic. Our republic located in far east of the Russian Federation on permafrost zone. As you probably know, permafrost is a natural freezer for the conservation of extinct animals. I am a paleozoologist and I'm studying mammoth faunous animals like woolly mammoths, woolly rhinoceros, cave lions, caribou, ancient bisons, and others. For every summer, our team works in the north of Yakutia for research of new finds of mammoth faunous animals. Our goal is to investigate a morphological study of ancient animals and find something unique of a previously unknown. At first time, I thought that if we found the most preserved specimen of an extinct animal, we would be able to bring it back to life by cloning. But unfortunately, a miracle is not happening. The fact is that cloning is only possible with living animal cells. Cloning is known to mankind for a long time. It is applied to plants and in the farming and is called cuttings. The principle of this method has been tried on animals and it's called cloning. The cloning process is quite simple. We have three sheeps, A, B, and C. From sheep A, collect a somatic cell. A somatic cell is a specialized cell that makes up the body, that is bones, muscles, fat, organs, and so on. From it, extract the nucleus containing DNA. From sheep B, collect egg cell. It is also extracted the nucleus. The nuclei of sheep A is placed into an unnucleated cell of the sheep B. And that cell is fertilized by a surrogate sheep C. From the sheep C is born clone of the sheep A. Currently, cloning method is developing intensively and it pleases. Since it is possible to reconstruct or correct the genome of an animal and cloning it, thereby giving rise to healthy and strong individuals of a species of an animals. Moreover, cloning could prevent the extinction of the modern animals. In case of ancient animals, the situation is quite difficult. Since in order to revive an extinct animal, need a confluence of many complex circumstances in time and space. For example, in order to clone an extinct animal, need a preserved chromosome. About the process of origin of life, death, and decomposition, in science knows a lot. For example, when a mammal dies, no matter from old age, accident, or disease, it began to decompose and rot. Then disintegrate into individual chemical elements. And it begins with the decomposition of the unit constituting the body of mammal cell. As you know, cell consists of organelles such as nucleus, mitochondria, Golgi apparatus, and so on. And all these organelles containing liquids of different composition are maintained during of life in working condition. That is, they are repaired, replaced, and updated. So, as a death of a mammal, all these processes stop, and the liquid contained in the organelle destroy the walls and mix together to form some kind of acid, which begins to corrode everything, including the cell nucleus containing information about the mammal. This entire initial decomposition process takes from 3 to several hours. Imagine the first case. Grazing woolly mammoths about 35,000 years ago, which from some reason falls dead. As you probably understand, the body will not be buried, and the irreversible process of decomposition will begin. The smell will attract scavengers and insects, and all these carcasses will rot and disintegrate within a very short time. That is, the genetics will have nothing. The second case, same picture, but in the winter time. The whole process of decomposition is also but a little slower. In both cases, the primary cell decomposition process will not change. And the third case, in nature there are natural traps in the form of bogs, marshes, and landslides. They existed in the late Pleistocene. So imagine a mammoth walking along its usual migration path, where after the last campaign, such a pit with liquid mud was formed. The case was in the late autumn, and a solid frozen crust formed on the pit. Mammoth unsuspectedly falls in the pit and dies. In this case, the animal, of course, gets into the anaerobic condition, and with the advent of cold weather, frozen in the ground, and remain so to the present days. But when released into the environment, the freezing does not take place immediately, and the primary decomposition process of the cells have time to complete, and DNA is not fully but partially destroyed. So, in the end, in order to clone, the same woolly mammoths needed instant stop process of cell decomposition by freezing. But in this case, there is one but. For example, if you leave a closed metal container with water outside in the winter, for example, in Yakutia, there are -30, -40 degrees, the water freezes and breaks it when it expands. The same happens with the mammalian cell. The water during freezing crystallizes and breaks the cell's walls from the inside, with high probability of destroying the nucleus with a chain of DNA. Thus, it is most impossible to find the surviving DNA chain of extinct animals. This conclusion can be drawn from the negative results of scientists who tried to do it with mammoth Zharkov in 1999, legs of Maksunocha mammoths, called by river, in 2002, the head of the Yukagan mammoths in 2003, the baby mammoth Luba in 2005, a mammoth from the island of Small Lechovsky, this one, and the mammoth Yuka in 2013 from the bank of the Laptev Sea. But in 2015, professor of Genetic Center of the Swedish Museum of Natural History, Luva Dalin, recreated the complete genome of woolly mammoths. But then again, we cannot use this result for cloning. The reason is that cloning requires intact chromosomes. But in Pleistocene samples, the DNA is fragmented into less than 50 million pieces. He can read the entire sequence by puzzling all the short sequences together on the computer. But there is no way to piece together the real DNA molecules. However, in the theory, one could use the nuclear genome sequence as a way to identify genes that are unique in mammals compared with elephant. Using this result, Professor George Church from Harvard can make synthetic copies of these genes and edit this into an elephant genome, cut out and then insert. His goal is to take the DNA responsible for the adaptive features of the evolution of woolly mammals to the cold climate and introduce them into the DNA chain of Indian elephant. Then by cloning to obtain a hybrid of hairy elephant. And if it will be possible, it is possible to resettle modern elephants which have reduced their habitat in other regions of our planet, thereby saving them from extinction. In conclusion, I would like to say that the arrival of animals that have become extinct in the distant past should take into account the ethical norms of our society. Since the revival of extinct or genetic change in modern animals could permanently affect our ecosystem, there may be a chain reaction of change in nature, which in turn can cause the death of other living animals and plants. For example, mammals were landscape-forming animals that are quite strongly influenced by the ecosystem of the past. After their extinct, the ecosystem has rebuilt, that is adapted to the absence of these species, and the ecological niche of mammals in the biosphere occupied by other species. And the appearance of a mammoth on the modern ecosystem can lead to catastrophic consequences for other animals and plants, and for the mammoth itself. This also applies to genetically modified animals. I am not saying that I am against cloning or against genetic or molecular engineering. On the contrary, I am for development of these areas. Scientists must bring this method to perfection because it has a powerful potential for improving the lives of future generations of people and animals of our planet. But at the same time, this area should take into account all aspects relating to the ethical standard of our society and characteristic of the modern ecosystem, which has changed dramatically since the time of mammals over the past 10,000 years, and it's most difficult and important in this case. And I want to talk about our new foundation in the permafrost. Last year and this year, this is Boris, his name Boris, because by the person who found it, his name Boris, and he gave it to it his name. And Spartacus, it was found, I think 2 or 3 months ago in August, and it's very good preservation, and I say the investigation is continuous. Thank you very much.